Hello, everybody. I know that things are a little bit crazy right now with having to switch over to distance learning and find new ways of doing stuff. And part of what that meant is we decided to spend an entire lecture just kind of making sure that everybody was on the same page. But in order for us to do that, that means that we're a lecture behind. I don't want you to fall behind in lab, and I don't want to start changing all of the deadlines in class because we're not expecting this semester to actually be extended. But you guys already know a huge amount about the cell cycle. In general, this is one of the lectures you guys like the most. It's probably one of the easiest, and there's a lot of resources for you that are available. So we're going to do this lecture via web campus videos. Please make sure that you watch all of these videos and that you sign into Socrative and follow along by doing the Socrative questions in the video as you go. I'll keep the video short, and you guys can come see me during office hours or talk to me more in recitation if you have questions. This is the information that's going to be in this week's SimBio. It's also going to be the topic that's discussed and practiced in recitation, and we're going to extend upon this in Thursday's lecture. So even though you guys do pretty well with it, please make sure that you don't skip on that preparation. Remember how hard it is when you really don't understand the foundations? This class is always about building on previous material. All right, so first I want you to pause the video here and do the first 6 questions in Socrative. When you're done, come back and we'll see how you did. Remember, you are not being graded on how accurate you are in Socrative. That is not the goal here. The goal is for you guys to test yourself and identify what you don't know. And if you don't know it and you get it wrong in Socrative, guess what? That means I also know that you don't know it. And that's how I know that I need to go back and help you out a little bit more. So there's no bad part here to getting it wrong. All right, so you did the first 6. Let's see how you did. Name the high free energy molecule that G protein coupled receptors use to activate their G protein. Well, G proteins are named because they use GTP as that high free energy compound that allows them to do work. Receptor tyrosine kinases are receptors that also act as enzymes to phosphorylate other molecules. Remember that these kinases actually dimerize. 2 RTKs come together, they phosphorylate each other, and that's how these complexes get activated and start downstream cascades. Name the enzyme that can remove a phosphate from a signaling molecule, reversing the signal. Well, if kinases can add phosphates, phosphatases can remove them. Cyclic AMP is an important small non-protein participant in signaling transduction pathways, also known as a secondary messenger. And it's going to be true that multi-step signaling cascades can be regulated or amplified at multiple stages. And it's also true that they're reversible. These are not all fits one kind of pathways. We can make changes to them. All right, so I want you to head back to Socrative, and I want you to take a look at this image. You're going to want to go ahead and do the next, I believe it's 4 questions, but just keep an eye out and tell me what type of microscopy was used to capture this image of the cytoskeleton. Then identify this cytoskeleton. So pause the video, come back when you're done. All right, so the first thing you have to decide is whether this is going to be light microscopy or electron microscopy. The fact that we see color tells us that this has to be light microscopy. And there are only 2 kinds of light microscopy responsible for knowing: bright field light microscopy or fluorescence. So this is an example of fluorescent light microscopy. So when you take a look at this and you think about this component of the cytoskeleton, well, you should recognize that this would be microtubules. And you might recognize that this is tubulin, which is the monomer that actually forms microtubules. These are organized at centers that are called centrosomes. And as a matter of fact, identifying and seeing these centrosomes within the cell as this central organizing center can help you to identify these as microtubules. Now, where would you find intermediate filaments in the cell? Hopefully you said circling the nucleus. Remember that nuclear lamina, which sits underneath of the nuclear envelope. And where would you find microfilaments, those actin filaments? Did you say in the cortex, that outer region of the cell? If so, you'd be right. Okay, I want to show you a video here that actually clearly demonstrates and represents a nicely dividing cell. So let me tell you what you're looking for. Shown in green here are the microtubules. This is what's going to be forming that mitotic spindle, the fibers that are responsible for separating sister chromatids. In red is the DNA. So at the beginning, you're going to see that DNA condensing down into chromatin, which you know as those little X's from all the cartoons in high school. And then you're going to see those spindle fibers pull those individual chromatids, those are replicates of the same chromosome, apart. And eventually you're going to see cytokinesis, where the cell actually divides into two separate cells. On exam 3, there's two different ways that we're going to ask you to be able to look at images and recognize them. The first would be images, microscopic images, maybe not always fluorescent, but different kinds of microscopy that are actually going to be showing you the cell dividing. And based on that image, the level of condensation, the formation of the spindle, and so forth, I'm going to ask you to tell me exactly which phase of mitosis or meiosis that you're seeing. The other thing that we're going to ask you to do is later down the line, when we actually tell you the number of chromosomes that are available in that cell, to look at those chromosomes and decide what phase you are in and whether it's mitosis or meiosis. So don't worry, there's going to be more practice on this coming, but just so you're aware, that's how you'd be tested. I want you to understand that it's essential that this process is completely ordered. If you were to go back and watch this video again, you would actually see one of those cells dividing into 3 separate daughter cells. That's because these are a cell line of kidney cells that are cancerous. And so that's actually a poorly regulated cell division caused by and something that's seen in cancer. And that's what you're going to be seeing and what we're going to talk about at the very end. If you properly regulate, maintain order and control, then the process of meiosis or of mitosis allows you to maintain the number of chromosomes and the genetic content in each individual cell. Now, most cell division results in 2 daughter cells with identical genetic information. In unicellular organisms, you can replicate an entire organism by using this process. Sometimes it's asexual reproduction, like binary fission in bacteria. But multicellular eukaryotes are going to use cell division, not only to replicate an entire organism, but also for development once an egg has been fertilized, organismal growth, and tissue repair. Now, I want to emphasize that first one to you. When a fetus is formed, when you actually get this fertilization of an egg, mitosis is going to be the form of cell division that's used to make that baby. The only purpose for meiosis is to form the original sexual gametes, the egg and sperm. Now, on Socrative, I want you to answer this question. Which of the following describes eukaryotic genetic structure? And if you need more time, go ahead and pause the video here. Hopefully this is a review for you, and you know that eukaryotes can have one or more, doesn't have to be one, okay, doesn't have to be more, but one or more linear chromosomes. Prokaryotes have a single circular chromosome. So the rest of this video is going to be about vocab. Much of it's vocab you've heard before, but most of them are going to be redefined and very clearly defined so that these words don't all start to blend together. You might imagine that the fill in the blank section on exam 3 is pretty big because you're going to see there's a lot of vocab. All of the DNA in the nucleus constitutes the cell's genome. So if you were a prokaryote, this would be all of the DNA found in the nucleoid. Some eukaryotes are going to organize their genome into multiple pieces. You are an example of this. You have 23 different chromosomes. Those individual pieces are what we call chromosomes. Each one of those chromosomes has different components. That's not to say that my chromosome number 1 is the exact same as your chromosome number 1. But all of the different types of codes, so all of the different genes, the fact that my chromosome 1 has an I gene means that your chromosome 1 has an I gene. That my chromosome 2 has a hair gene means that your chromosome 2 has a hair gene. Doesn't mean that we're going to have the same versions. We're going to define that in more detail coming up next week. The number of chromosomes, the number of copy of those chromosomes is also important. I don't have just one copy of chromosome 1. I have 2 of them. I actually have 2 genes that talk about my eyes. One that I got from my dad and one that I got from my mom. So humans are going to be what we call diploid. So diploid organisms have 2 copies of each piece of their genome, meaning 2 copies of each chromosome. If you are triploid, you'd have 3. If you are quadruploid, you'd have 4. If you are octoploidy, you'd have 8. That's a strawberry, by the way. So even though each chromosome has the same structure, it has the same composition, there can be different versions of them. Eukaryotic chromosomes are also associated with proteins. And this is important because these proteins not only help to organize this information, but during the process of cell division, when the giant genome and all those different pieces have to be organized, sorted, and separated, histones allow us to condense those very, very long linear strands of DNA down into a very condensed structure known as chromatin. In non-dividing cells, the DNA is going to be less condensed. So we're going to have a less condensed version. Those canonical X's that you're used to seeing as a way of representing chromosomes, that's really this condensed chromatin version. So if you take a look here on the slide, you're going to notice what these chromatins actually look like. Let's see if I can get my little spotlight up. Aha. So here's the center of your X. The X is actually one line and a second line when you would draw the X. This entire thing here is a chromosome. The chromosome has been duplicated though, in preparation for the cell to divide, right? If you're going to make 2 identical cells using the process of mitosis, it's going to be important that each one of them has an I gene. So you need to duplicate that I gene by duplicating the chromosome so that each of the daughter cells gets to keep its copy. We call these duplicates chromatids. And because they're identical, we call them sister chromatids. So sister chromatids are simply the 2 copies of a chromosome, and they're attached. This central part here, center, centrosome, okay? I'm sorry. This center part here is a centromere. See, even I do this. There are centrosomes, which are central centers. That's how I like to remember it. Central centers for organizing microtubules. Centromeres are centers of chromosomes. See that M? So this centromere is the central region of this sister chromatids. I know it's a little confusing, and then now I have to tell you that not only are chromatids duplicated chromosomes with a central region called a centromere, but they're held together along their length by yet another C word known as cohesins. If you remember the word cohesive, well, these are proteins, cohesins, that are going to hold together the two sister chromatids. So the protein complexes that are here in the middle are called kinetochores, and these are going to be another set of proteins. But kinetochores are kinetic, right? Like kinetic energy. And what you're going to learn is that these kinetochores are important for the movement of proteins. Cohesins hold them together like a cohesive force, but kinetochores actually help to move them. So chromosomes, which in this case have been condensed down into chromatin and are shown in blue, are attached to kinetochores, shown in green. These are located in those centromeres, the central regions of the sister chromatids. And they're attached to microtubule spindles, which are shown in red. And these fibers allow these kinetochores to walk each one of these chromatids along the spindles, and it separates them into each of the two daughter cells. So now I want you to get out your Socrative. There's a lot of vocabulary that's going on here, and the more we use it, the more comfortable you're going to get with it. So why don't we just start by using some of it? Tell me in Socrative, when DNA is condensed, what do we call it? Good, this is chromatin. Now, chromatin is condensed because the DNA is associated with proteins called what? Hopefully you recognize these are histones. Now, each piece of DNA in the genome is called what? So each individual piece of DNA. Well, you have 23 of them because you have 23 chromosomes. But when we duplicate each one of those chromosomes, what do we call those two copies? Ah, yes, these are chromatids, sister chromatids. And that waist here that's attaching, that's holding these sister chromatids together in this chromosome, that central waist, what do we call that? Yeah, that's the centromere. Now, not shown in this picture, you'll notice that they've gone back to this canonical X formation, where they show these sister chromatids with waving, wiggly arms. But it turns out that's not the case. These sister chromatids are being held together along their length by proteins, and those proteins are called cohesins. Remember not to get centromeres confused with centrosomes. Centrosomes are that central organizing center used by microtubules. And if you don't remember what they look like, go back to the start of the video, where I showed you a picture of those cells. All right, here's the end of video 1. If you got some of these questions wrong, if you're not entirely sure about what I'm talking about, or if you're just confused about the vocabulary again, well then take a break, go through your notes, maybe pull out your textbook. That's really good for reviewing vocabulary after all. And come back to see me again when you're refreshed for video number 2.